Hi guys, this is Brian. I'm going to show you today just a little bit about what we'd be doing if we were meeting face to face, which is to look at mosses using microscopy. I'm sorry that we can't do this since we're online this semester, but eventually perhaps you'll get the chance to take a moss indoors and look at it under a dissecting scope and a compound scope and use the real keys to figure out what you're looking at. That's the only definitive way to tell many mosses apart, because they are just so tiny that you can only see so much with the hand lens. So I'll first show you how to collect a moss, and then we'll take it inside and I'll show you what to do with it after that. So I'm out in my yard and I have mosses everywhere because I don't do much in the yard. I just let it sort of grow. Down here by some ferns I have mosses in the ground. The majority of these are atricum, though there's some other things. So I'm going to come in with a knife and just loosen the soil around some. And the idea here is to try to get the plants without breaking them apart too much. And you don't need a lot. So about a $50 sized piece is enough, and you can shake off the soil. So what I have here is probably 15 or 20 stems of atricum, and I believe this is probably atricum ultracristatum. And we'll take this inside and pick it apart under the scope. So this is my home lab, and I have stuff mostly for looking at grasses and sedges, but also mosses and liverworts. And I'll be starting to study liverworts in here in more depth next week as part of a course that I'm taking. So I have a dissecting scope, and it doesn't have to be anything fancy. This happens to be a pretty nice one. And a compound scope, which is necessary to look at the leaves more closely. So you really need both of these. I have my manuals. These are the ones I like the best to start with. Sue Williams' Ecological Guide to Mosses of New England, and Jerry Jenkins' Northeastern Mosses, a Graphical Guide. And there is a copy of this as a PDF on the Brightspace site for you to download if it would be of use to you. Other tools that I need are water, just dropper water, and then fine needle forceps, which I protect by keeping them stuck into a wine cork. And that's really all you need. You need paper and pen to take notes. And what I'm going to do is try to set the camera up so you can just see me working. And then I've got some other videos that I've shot that I'll try to intersperse as I put all this together. So I have my scope and the camera set up, and it's not my best camera, but that's all I can do at this time. So getting organized, just clear some space. I have the moss that I picked out in the yard, and I just set that on a simple tray, which in this case is the top of a freezer container. Get your slides and cover slips together if you haven't done that already. And I just keep some Kim wipes around, or tissues to wipe slides and such with. Use a little water if you need to clean a slide off. Very simple tools. And then clean the slide off. Always better to do it beforehand, but this is what I had. Okay, so I've got slides and cover slips ready to go. So now, working under the scope, you just pick your moss apart. As you can see right here, just pick everything apart and lay it out so that you can work on it. And then get your forceps and start working under the scope on the lowest magnification to isolate a couple of branches, which you'll then use to isolate leaves from. This short video clip shows the isolation of a shoot of Hypnum imponens, which is a pleurocarp type moss. But you can see the process of pulling moss apart under a dissecting scope. And as you can see, the majority of this is not going to be used, so I lay that aside. Now, I've got my leaves there, and just start with one short little stem at a time, and there's not much to it, just a tiny little piece. And that's why you don't need to take a lot of moss. And you start up working dry. This video clip shows the isolation of leaves, again from Hypnum imponens, which I did earlier. So this will be different than the atricum, but the process is the same. You clean up your sample, and you then slide in and pick off as many leaves as you can. With this particular moss, it's easy just to go in and pull the tweezers right down the stem, and that pulls a whole bunch of leaves off all at once. And then in water, you spread those leaves out before you put on the cover slip. It's important to clean the garbage out, the large material, because otherwise the cover slip will not sit flat against the leaves and you'll have trouble getting flat samples. Maybe moisten it a little bit to get the leaves to unfold. And just look at what you have. I can see in here that this is an acrocarp type moss. It has a strong central costa with lamellae. That's how I know it's an atricum. The leaves are wavy towards the tip and strongly toothed. So this might actually be Atricum undulatum, but I won't know until I look under the microscope. And then working under here, and there'll be a separate video showing this, you come in and basically hold it and peel the leaves off and make a pile of leaves on the slide. And if you're having trouble with the leaves sticking to the forceps, you can actually put a drop of water on the slide, and that has enough of an attractive force that everything will stay within that water. If you're really good at this, you can actually grab the tip of the moss and just slide the forceps down to peel off a whole bunch of leaves at one time. And I have a video of that you can see. And so how many leaves do you need? The more the better, but you don't need a ton. So something perhaps like at least 5 leaves and maybe as many as 20 leaves would be a good start. And then you lay these leaves out under the scope so that they don't overlap, so that when you put the cover slip on, things will be sitting well enough apart that you'll be able to get good views. And at this time, with the leaves in the water, you can take another look, and you can see different features. You can change the magnification. This video clip shows a cover slip being put into place, and if you have too much air under the cover slip, you just add a little bit more water to the edge and it'll get sucked right under the cover slip. Once we focus into these Hypnum leaves at a higher magnification, you can see the alar cells at the base of the leaves. Those are those reddish brown cells at the base, and they're involved in leaf movement as the moss hydrates and dries out. So we are isolating these leaves of Polytrichum commune, the common haircap moss, because we want to section them crossways so that we can view the lamellae, or small stacks of photosynthetic cells, on the surface of the leaf, which is part of the process of identifying this moss to species. So I'll put the slide across to hold them in place, and then I'll cut them with a razor blade as I gradually draw the slide back to make small, thin sections. And these then get put in a drop of water and looked at under the compound scope. And hopefully some of them are going to sit sideways so we can see the lamellae, and I have a picture of this later on. This is the same technique that you would use to section atricum leaves to look at the lamellae, and the height of the lamellae and number of lamellar rows are key to identification of atricums to species. I can see here that this has low lamellae on the upper surface of the midrib. I can see the toothing, I can see the strong costa. I can see the base of the leaves, but I can't really see if there are any alar cells. And then you take this and put a cover slip over the top of it. Push it down to exclude any air bubbles, and then you're ready to take this and put it on the compound scope over here. And again, you start under the lowest magnification, and you come in and move around and start seeing what there is to see and take notes. So this is a water mount, and you get a much better view of the margin of the leaf. You can see whether there's a bordered margin and whether there are what the teeth are like, whether they're single teeth or double teeth. You can see the midrib very clearly. I can see that there's 4 rows of lamellae that look to be about 4 or 5 cells high. So that's part of the identification. To tell this atricum apart from other atricums, it's easy enough for me to tell that this is the genus Atricum, but to get down to which one it actually is, you have to do this. This shows a leaf of Atricum under the compound scope, and you can see the lamellae standing on top of the costa in the middle of the leaf. This picture is of Polytrichum commune under the compound scope, and this is a cross-section of the leaf, and you can see the stacks of lamellae that are all over the surface of the leaf, not just on the midrib. So now we're looking at leaves of Hypnum imponens under the compound scope, and we'll look first at low magnification, and you can see the alar cells, and you can see the cells in the leaf itself. You can also see that the leaf is ecostate or has no central costa in this case. Other things to notice here are that the alar cells are colored and inflated at the base, and then also the margins of the leaf are entire except near the tip of the leaf where they're slightly toothed, and the tip of the leaf curves off to one side and comes to a sharp point. Under the next level of magnification, you can see that the cells in the leaf of Hypnum are elongated or worm-like. These are all diagnostic characteristics. So this is Mnium hornum, the lipstick thyme moss seen under the compound scope. You can see on this leaf that it has a strong central costa, that the cells of the leaf are round, and that the edge of the leaf has a strong border, and that border is toothed. Under the next higher level of magnification, you can see that the teeth on the border are doubled up, so it has double teeth, and that's a characteristic of this particular species. And this can only be seen under the higher magnification. Mosses are really beautiful under microscopes, and because they are only one cell thick typically, the light transmits through them and you can see chloroplasts, cellular structures, and the diversity of teeth and cell sizes and shapes, borders, and all sorts of things. So this is a leaf of Thelea hirtella, the train tracks moss. And what I want you to look for as you watch this video is how the leaf cells are papillose. They have bumps all over them, very intricate sometimes. And then also in this slide you'll see little pieces of green linear tissue called paraphilia, which are small hair-like structures that are in the axils of the leaves, and they break off and form new mosses. So this is a method of vegetative propagation among mosses. So I'll look at this, I'll take notes. I may actually decide to go back to my dissecting scope and section the leaves crossways, and there's a short video of how to do this that will be part of this, and then I'll put it back under the compound scope to take a closer look. So you would have been doing this if we had done this in person. I would like to offer to any of you the opportunity to learn more about this if you're interested in the spring. You could either just arrange time with me to do it, or you could actually do a special problems course and get a few credits for getting more in depth with mosses and liverworts throughout the semester. Okay, so I'll break this off here and then try to go put a video together for you. Thanks for joining me in my lab.